Good morning all. So today we are going through the continuation of Krebs cycle. Actually we have discussed up to this point, that is the pyruvate in the presence of carbon dioxide, which may result in the presence of an enzyme pyruvate decarboxylase. This pyruvate, which is produced from the glycolysis, it can be converted to acetyl-CoA plus NADH plus carbon dioxide. So, the pyruvate can be converted to acetyl-CoA in the presence of the enzyme pyruvate decarboxylase, and as a result we get acetyl-CoA along with NADH and carbon dioxide. The product of this reaction, that means acetyl-CoA, it is the starting point for the citric acid cycle, and this acetyl-CoA may also be obtained from the oxidation of fatty acids. Apart from pyruvate, it can also be obtained from the oxidation of fatty acids. So this is the diagram that I have already discussed yesterday, that is oxaloacetate is acetyl-CoA, which pyruvate can't enter the Krebs cycle, so it can be converted to acetyl-CoA. Then it enters the Krebs cycle combined with oxaloacetate. Then the cycle of intermediates are formed, etc. The citric acid cycle, and it may begins with what? The transfer of a 2-carbon acetyl group from the acetyl-CoA to form 4-carbon acceptor compound, that means oxaloacetate, to form a 6-carbon compound called citrate. This is the 2-carbon compound, and this can combine to form a 4-carbon compound, and this both acetyl-CoA converted to this combined with oxaloacetate, it may result in the formation of a 6-carbon compound, 1, 2, 3, 4, 5, 6, that means citrate. And the citrate then goes through a series of chemical transformations, losing 2 carboxyl groups as carbon dioxide. So it lost 2 carboxyl groups. The carbons lost as carbon dioxide originate from what was oxaloacetate and not directly from acetyl-CoA. So as a result of the losing 2 carboxyl groups, citrate moves through a series of chemical transformations, and it may reaches up to oxaloacetate, which is it can be converted to a 4-carbon compound, oxaloacetate. The carbons donated by acetyl-CoA becomes the part of oxaloacetate. I have mentioned this point earlier. That means acetyl-CoA, the carbons of acetyl-CoA combine with oxaloacetate carbon backbone after the first turn of the citric acid cycle. So the loss of acetyl-CoA donated carbons as carbon dioxide requires several turns of citric acid cycle. So it requires what? Several turns of citric acid cycle. However, because of the role of the citric acid cycle in anabolism, they might not be lost, since many citric acid intermediates are also used as precursors for the biosynthesis of other molecules. So, in the cycle reaction, there are many intermediates are formed in the Krebs cycle. So, these intermediates can individually used as precursors for the biosynthesis of other molecules also. Most of the electrons made available by the oxidative steps of cycle are transferred to NAD+, forming NADH. So, the most of the electrons can be transferred to this coenzyme NAD+, and it can be converted to NADH. For each acetyl group that enters the citric acid cycle, 3 molecules of NADH are produced. So, in the overall reaction, 3 molecules of NADH are produced as a result of the transfer of electrons to NAD+. Okay. Then the citric acid cycle, it induces a series of oxidation reduction reaction not only in the prokaryotes but also in the eukaryotes also, that is within the mitochondria. For every NADH and FADH2 that are produced in citric acid cycle, 2.5 and 1.5 ATP molecules are generated in the oxidative phosphorylation that we will discuss in later. That means electron transport chain is oxidative phosphorylation. So at the end of each cycle, the 4-carbon oxaloacetate can be regenerated. So again, after combining this acetyl-CoA with oxaloacetate, it can be converted to citrate, isocitrate, alpha-ketoglutarate. Like that the cycle goes on. Finally, it may regenerate what? Oxaloacetate. And again this cycle is continuing. That is the process of Krebs cycle. So steps. So there are 10 basic steps in citric acid cycle as outlined below. The cycle is continuously supplied with new carbon in the form of acetyl-CoA, that is entering the step 0 in the table. So through this acetyl-CoA, this new carbon can be continuously supplied throughout the Krebs cycle in the form of acetyl-CoA. So these are the different steps of the citric acid cycle. First is this oxaloacetate plus acetyl-CoA plus water in the presence of enzyme citrate synthase. It gives the product citrate plus CoA. CoA is from this acetyl-CoA. Then SH, CoA SH. And the reaction occurring is aldol condensation. It is irreversible. Extend the 4-carbon oxaloacetate to a 6-carbon citrate molecule. Next step, the citrate which can be converted with the help of the enzyme cis-aconitase. And this can be, it is a dehydration type of reaction that removes water and it may result in the formation of cis-aconitate from citrate. And it is a reversible isomerization. It can be reverted at any time. Again, this cis-aconitate, by the removal of the water, it can be with the help of the same enzyme aconitase. Now it is a hydration reaction. It can be converted to isocitrate. And this is also reversible isomerization. So citrate to aconitase, cis-aconitate, then to isocitrate. Third step, the isocitrate molecule in the presence of the enzyme isocitrate dehydrogenase. It is a type of oxidoreductase enzyme. And the reaction taking place is oxidation of isocitrate to form what? Oxalosuccinate. And because of this dehydrogenase enzyme, electrons are transferred from NAD plus to NAD plus. This may result in the formation of NADH plus H plus. So the product in the third step is oxalosuccinate, and it is simply an oxidation reaction. And in this step, it generates NADH. Look, this is the NADH, and it is equivalent to 2.5 ATP. Next last step is, fourth step is oxalosuccinate. This oxalosuccinate again can be converted with the help of the same enzyme isocitrate dehydrogenase. Now this time it is a decarboxylation reaction. This converts oxalosuccinate to alpha Kg or alpha ketoglutarate, and the carbon dioxide is liberated. And this step is important because it is a rate limiting step and also it is a irreversible step in glycolysis. It generates 5-carbon molecule. Next is fifth step, that is alpha ketoglutarate or alpha Kg with the help of the enzyme alpha Kg dehydrogenase, thiamine pyrophosphate, lipoic acid. It is a type of reaction called oxidative decarboxylation, which may result in the formation of succinyl-CoA plus again because of the dehydrogenase enzyme, it may transfer electrons to NAD+, it may form NADH plus H plus CO2. Here oxidative decarboxylation takes place, and this step is also irreversible stage. It generates, here also NADH is generated along with succinyl-CoA. It regenerates the 4-carbon chain. Next is succinyl-CoA plus GDP plus PI. So already the GDP, energy requiring molecule, it combined with succinyl-CoA. So succinyl-CoA plus CoA SH GTP, CoA SH GTP, it is the product which is formed from succinyl-CoA enzyme succinyl-CoA synthetase. So product is succinate plus CoA and SH are removed. It is a type of substrate level phosphorylation. So we will learn detail about both substrate level and oxidative phosphorylation in another video. So this much you study the production of ATP from the substrate itself. So because of this succinyl-CoA plus GDP, succinate is produced with the production of GTP, or this ADP can be converted to ATP. Instead of GDP to GTP, it generates one ATP or equivalent condensation reaction of GDP plus PI. So it generates one ATP or equivalent condensation reactions of GDP plus PI. And the hydrolysis of succinyl-CoA involved the water needed for the balanced equation. Next step is succinate. The product succinate plus the ubiquinone. It is a type of intermediate in electron transport chain, ubiquinone. With the help of succinate dehydrogenase, it may form a fumarate plus ubiquinone. Fumarate plus here also an oxidation reaction occurs, and here what happens is that FAD acts as a prosthetic group which can be converted as a result of this oxidation reaction. FAD can be converted to FADH2 in the first step of reaction in the enzyme. These two electrons are later transferred to QH2 molecule. So what happens in this step? This FAD can be converted to FADH2 in the enzyme. These two electrons are later transferred to QH2. This is ubiquinol during the electron transport chain, where they generate equivalent of 1.5 ATP. Next is, so what is the last product which is formed from succinate? Fumarate. The fumarate, in the presence of enzyme fumarase, which is a hydration reaction, it forms L-malate plus, and the reaction is hydration. The next is, so the hydration of CC double bond occur. Next is this product L-malate can be converted to oxaloacetate, and here also malate dehydrogenase enzyme which may result in the transfer of electrons. This oxidation reaction occurs and NAD plus convert to NADH plus H plus. It is a reversible reaction. In fact, equilibrium favors malate and it generates NADH. Finally, this oxaloacetate which reacts with acetyl-CoA plus water. So again, the incoming acetyl-CoA from the glycolysis, which has been converted from pyruvate or oxidation of fatty acids, this oxaloacetate and acetyl-CoA combine together with the help of the enzyme citrate synthase to form citrate, and here same aldol condensation occur. This is the same step as step 0 which we have mentioned earlier, and it restarts the cycle. The reaction is irreversible and it extends the 4-carbon oxaloacetate to 6-carbon molecule. So these are about the important steps in the Krebs cycle. In the next class, we can discuss the regulation and different